Alright, my AP biology students. Today we're going to talk about eukaryotic gene control. Eukaryotic cells are more complex, consequently, so is the regulation of their genes. And since eukaryotic cells are more advanced evolutionarily, that means that there tends to be a lot of inefficiencies and almost wasted DNA inside their genome, which of course makes controlling it require a more diverse group of processes. All right, so the first thing we need to talk about is how DNA is organized. DNA is organized into, chromosomes, which we talked about during mitosis and meiosis, and those chromosomes are used for the process of replication. Now, those chromosomes during interphase are in this sort of uncoiled string called chromatin. Well, there's basically two types of chromatin. First, there's heterochromatin, which is chromatin that's tightly packed and it's just being stored, it's not being used. And the other type is euchromatin, DNA that's being transcribed. It's DNA that's currently actively being used. It tends to be more loosely packed, that way all of the structures can get near it. And what we found is there's actually some pieces of it that are sort of attached to the inner portion of the nuclear membrane, or the nuclear envelope, and that sort of allows for control of transcription. It allows certain sections to happen faster, certain sections to happen more efficiently. So that's the sort of big structure level of control. Now, to get at some of the other aspects of control, we need to talk a little bit about how genomes are organized. All right. One of the things you have to understand is that eukaryotic genomes contain mostly non-coding sequences. They mostly have sections of DNA that don't really give you any proteins, and those sections tend to be regulatory. We've talked about that a little bit in terms of operators, promoters, that kind of stuff. They tend to be introns. Remember, sections of the DNA that are coded into messenger, pre-messenger RNA are then excised. The exons are joined together, the introns are removed. Well, that's sections of the DNA that don't give us a functional protein. And then there's repetitive DNA sections, which we call satellite DNA. They tend to be like 10 to 15% of your DNA, and they're just highly repetitive short sequences. They're non-coding, mostly just structural, to make sure that as these processes that we use, like replication and mitosis, aren't actually destroying the DNA. That's why you tend to find them mostly in centromeres and telomeres near those sections. We also have sort of interspersed repetitive DNA, all right, and that tends to be 40% of mammalian DNA, and they all seem to have similar sequences, but they're hundreds of thousands of base pairs long. Most of them are transposons, pieces of the DNA that have been moved from one section of the chromosome to the other. That creates variety, but it also sometimes creates large pieces of repetitive uncoding DNA. Also important to bring up is that some genes are families. All right, you have to understand most of your genes are unique, one gene, one protein, but some genes are related. They're very similar in structure. Sometimes they combine. A great example is hemoglobin, which has 4 very similar chains that combine to produce a overall hemoglobin molecule, and all of those are from the same family. They're slightly different. Sometimes it's because of a common ancestor factor. Genes have varied through generations, but we've still maintained all versions of it. Usually gene families tend to code for RNA products. A lot of those are used to code for rRNA. All right, so most genes are one gene, one protein, but there's sometimes are multiple families where they're similar, they're related, and sometimes they give you one protein together. There's also this thing called pseudogenes. They're actually functional protein structures that don't code because they don't have the proper regulatory sequences in front of or behind them. And so while they could potentially code for an appropriate gene structure, they don't, because they don't have anything to turn them on, so to speak. Now, you have to talk a little bit about some of the things that we can do to alter a genome, because when we alter a genome, that can frequently change the way that it's coded. All right. First off, you can have what's called gene amplification, where sometimes a section of a chromosome ends up being replicated, and that causes the gene to be overproduced. All right. So that's something that can occur. Also, there's transposons, which is what this diagram is on the left. Basically, what you have with transposons is you have have a section of DNA, which is what's coded for right here. And in the presence of certain enzymes, certain proteins, this transposon will actually insert itself in this section of the DNA. And what that does is that takes this and this, and it can actually bring them together and shorten them. It doesn't always have to. And then also, what it does is it takes these purple sections, which were a gene, and now just inserts a DNA segment in the middle of it, which drastically alters this gene because it changes the amino acid sequence, which then changes the structure, which then changes the function. All right, but this creates variety. All right, which could be good or bad. There's also a special type of transposon called a retrotransposon, and those are actually a sequence of transposons that create reverse transcriptase and insertion enzymes, which actually help the transpositional process. Final thing we want to talk about in terms of alterations to a cell's genome, and these again are just the most common things, is immunoglobulin alteration. Immunoglobulins, antibodies, they are proteins that are produced by B lymphocytes, which are B-type white blood cells, and they bind to infectious agents, proteins, viruses, bacteria, and they signal your immune system that, hey, come get this, come destroy it. Well, immunoglobulins have to be subject specific. You have to have an immunoglobulin for, one type of the common cold. You have to have an immunoglobulin for the other type of the common cold. You have to have an immunoglobulin for, a streptococcus bacteria. You have to have an immunoglobulin for, the flu. And so to create that sort of variety, your immunoglobulins are actually hundreds of regions of DNA that during the process of B lymphocyte differentiation are randomly combined. And that creates new, different varieties. And then when you need those antibodies produced in mass, that one cell divides and produces them. That's actually why it takes you a couple days to start fighting off an illness. So let's talk a little bit about some of the ways these genes are regulated. Let's start at the chromatin level. All right, first off, you can actually have enzymes that add methyl groups, which is a CH3. Okay, so a methyl group is a carbon with a hydrogen, and a hydrogen bound to it. Okay, and methyl groups are something that they get added mostly to cytosine on the DNA chain, and they actually prevent transcription from occurring. All right, it doesn't allow the DNA to be accessed appropriately. So that's one way you can shut it off. But in addition to shutting it off, you can also speed it up at the chromatin level. And that's where this histone acetylation comes from. Acetyl groups, similar to the methyl groups, they involve double-bonded oxygen and a couple other structural components. And what happens is they're added to certain amino acids on histones. And what that does is that sort of opens up the DNA that's currently wrapped around histones and sort of creates that euchromatin structure, which allows easier access for transcription. All right. Interestingly enough, the transcription factors that are required to allow the process to begin tend to increase the process of acetylation. And enzymes that methylate DNA also tend to deacylate histones. So you've got these enzymes that sort of stop the process, make it slower, and these enzymes that speed it up and make it quicker. And they all tend to work together. Gene regulation also, promoter sequences that sort of get this whole process going, show where transcription factors, where to bind, that kind of stuff. Introns, removing the exons, that kind of stuff. It all helps regulate it, because remember, regulation means making it happen, and it also means making it stop. So those things all are, keeping it happening at the right pace. And so these things all contribute to that. Transcription factors, whether they're present, how easily they bind, whether there's enough, whether they bind and then unbind, whether they lock down, whether you inhibit them allosterically. All right, all of those things can help transcription factors control the process of transcription. There's also enhancers, which are sections of DNA. When it says distal control elements, it actually means sections that are downstream. They're not necessarily in with the gene. And what happens is the DNA sort of flexes over, and the enhancer helps the transcription factors stay in place. Also, this is sort of a steroid thing, but steroid control molecules that bind to the DNA to help the process of replication, help the process of transcription, translation. They're actually molecules that bind to multiple promoters, and that's actually sort of uncommon. And then this diagram right here, which sort of went over that. you'll notice, see how it has the enhancer region labeled right here? Okay, this enhancer region is a section of DNA that binds this protein, which helps hold a transcription factor in place and helps the process of transcription occur. So again, like I said in the beginning, there's a lot of processes involved in regulating gene expression, and it happens in a lot of places, including after it's been transcribed. You know how introns and exons are removed? There's actually alternate methods for that. So sometimes you can remove different sections of pre-mRNA transcript to change the structure. Also, you have to think about this. Messenger RNA doesn't last forever. It's not meant to. So it breaks down pretty quick, and that, stops the process of translation. Well, how quickly is determined by the poly A tail. The longer the poly A tail tends to be, the longer the mRNA lasts before it's degraded by nucleosomes, which are enzymes that destroy nucleotides. Also, the 5' cap, all kinds of proteins, structures bind to that can either slow down or speed up the process of translation. All right. So again, we can do this at the chromatin level with methylation. We could do this with promoters, enhancers, introns, but we can also do this during the translation level by either breaking down the mRNA, stopping it from being broken down, using proteins to induce translation, or using them to stop translation. Also, you can even just destroy the protein. Proteasomes are structures that degrade proteins which are marked for destruction. Now, the last thing we want to talk about here is the whole sort of cancer thing. Because, a lot of what goes on in cancer, the unregulated division of cells, happens on a chromosomal level, on a genomic level, on a DNA level. And they're these things called oncogenes. All right? No, you don't have genes that give you cancer. We call them oncogenes because they're genes when there's alterations made to them, then they cause cancer. All right, and they tend to be alterations that can cause uncontrolled cell growth. So they tend to be involved in things like the mitosis promoting growth factor, or in kinases, or the cyclin dependent kinases, or the cyclins. All right, so the things that produce the cyclins, sometimes it makes them too much. The things that react to the cyclins, sometimes it produces them too much. The kinases that start the process after the cyclins bind to them, sometimes it makes them too much. All right, that's what sort of happens here. And these changes in oncogenes can come from a retrovirus inserting its DNA. It's actually what happens a lot in our concern with human papillomavirus, all right, HPV. Those viruses actually can insert their DNA into your own. They can damage certain genes, which can create oncogenes. Transposons can actually change a gene. We talked about that. Or they can actually move a gene from an area where it's being regulated properly to another area where it's not being regulated at all. All right. Or if it's a gene that slows down cell division, they can move it to an area where it never gets turned on. All right. There's a lot of things that can happen. Now, there's two genes that are involved in a high percentage of cancer cases. I just wanted to bring up. One of them is called the RAS gene, and it actually codes for a receptor on the cell membrane that reacts to growth factors. And a very common mistake in that is that it changes shape, so it reacts as if something signaled it to grow, but nothing did. There's also a problem with the P53 gene. Now, the RAS gene is an example where a change has occurred that causes the cell to divide more frequently. P53 is more of a regulatory gene. It's a gene that codes for proteins that stop growth proteins. It also is responsible for initiating apoptosis, controlled cell death, if there's reading errors when DNA has been replicated during the S phase of cell division. So you get two different examples there. I mean, and there's a bunch of others. I know one of the research projects that we did was on the BRCA, which is a breast cancer gene, regulatory gene. So there's lots of different examples. One of the last things I want you to walk away with is the understanding that cancer is a multitude of genetic regulation errors. One will usually not cause it. Your cell will catch that and destroy it. It has to be a multitude of mistakes that lead down that road. All right, looks like we're done for today.